Thanks. First of all, thanks, Philippe, for organizing this. I think it's a wonderful session. And yeah, good to have more structure in ISMB, more than 3D6. So good to have an extra day of that. So yeah, I am Jose Duarte from RCSB Protein Data Bank in UCSD. And I'm going to talk about RCSB.org, which I suppose this audience knows quite well, but anyway, I'll get into some of the latest developments. First of all, some context. The PDB itself is the single global archive for experimentally determined 3D structures of biomacromolecules. And it was founded in 1971 with 7 protein structures. Here we show 4 of them. And now we have 220,000 plus after more than 50 years. And so there's been formidable growth in experimental knowledge. And it was the first open access digital data resource in all biology. So it was really pioneering the open access movement much before it even existed. So then on how it is managed, there is a framework organization there, WWPDB, Worldwide PDB, that sort of is a framework for all the regional centers. So we have in the United States, it's us, RCSB PDB. If you're wondering what that means, it's Research Collaboratory for Structural Bioinformatics. And it is the archive keeper, and it is the direct successor to the Brookhaven original site. And then we have in Europe PDBE, and then PDBJ in Osaka University in Japan, and more recently PDBC in Shanghai, China. And then we have associated data resources for electron microscopy data, EMDB, and for NMR, BMRB. And then how it works, the different regions are depositing through the different centers. And then that's on the data inside, on the deposition side of things, and then on the data outside, we have independent offerings. And RCSB.org is the offering from the US organization. With RCSB.org, you can browse, visualize, compare, and search, all of it in a single web resource. And then I want to talk also about, of course, AlphaFold and revolution that came with it. We call the predicted models from AlphaFold compute structure models. And well, we've heard a lot about it from John and from other people this morning. I like this phrase I heard at CASP from Nick Grishin. I think that he said that what AlphaFold is showing that is the structure prediction problem is solvable. So basically, it did very well when it was presented in 2020. It's not solving the whole thing, but it went a long way to solve it. And we are seeing every week almost the new developments that are showing that things can be solved. The whole problem can be solved. And importantly, in 2021, the software was released to the public, and that allowed the whole community to just play with it. And then AlphaFold DB came as a collaboration between PDB Europe and DeepMind. So with that, it was clear that we had to acknowledge that the importance of these models, and then integrate those models into the RCSB.org resource. So that's what we did. This has been online since 2022. We have more than 1 million computer structure models from AlphaFold DB and Model Archive, basically covering all the model organisms and some others. And basically with that, we can call it a one-stop shop for the whole of biology. It's also not only the experimental structures, you can also see the computer ones together and do stuff with them. So let's see what you can do. You can browse. I'm sure you use our website. You're familiar with the structure summary pages that have summaries for each of the experimental structures. But now you also have summaries for each of the CSMs that we are integrating. They are very clearly differentiated with icons, with different warnings for the computed ones, and with a prominent display of all the confidence scores, PLDT. And then you might not be so familiar with this one. This is more recent. This is a group summary page, which is a very useful way to browse through proteins of the same, a group of proteins that are have a certain similarity, for instance, sequence similarity, some sequence cluster essentially, and you can browse through them and very quickly look at the different members of this group and filter by different criteria, etc. You can visualize, and I want to show this example of the sequence annotation viewer and Monstar viewer that we offer as a single application and interactive. Maybe just well, we are in the visualization session. We've heard a lot about ISMB. Monstar was originally created by a collaboration between the RCSB PDB at UCSD and the PDB Europe and David Senel at the Central European Institute for Technology in Czech Republic. And it's been since, it's grown a lot, and it's been still maintained by PDBE, by a larger community, and partially by us too. And on the left side, we have the sequence annotation viewer, which was created by Joan Segura at the UCSD site too. And this is sort of something we put together as part of our RCSB.org, but they are independent applications that can be used in your own programs if you have your own websites. So that's something that you can do too. Comparison. This is another example of these two applications working another way. You can do comparisons, and that includes in this group summary page, you can do comparisons of AlphaFold models and experimental ones. And last but not least, search. Search is really very important, especially with now 1 million models and more to come. You can do searches that you can combine scientific searches like structure searching, the 3D, matching the 3D coordinates with text searching. So you can, for instance, mix this structure search with organism search, which is a textual annotation. And that's based on a method developed in-house, the structure search, called Biosarnike, was done by Dmitry Busenko, a postdoc at UCSD. And now we are working on a new. It has some problems on matching, local matching. So I can only do global kind of matching. So with what we're developing now, that's Joan Segura and UCSD. It's a deep learning based method that can do local matching as well, and it can be scaled to any number of structures. And behind all that, there's a lot of web services, APIs. Now we have made them discoverable by putting these cogwheel buttons throughout the website. So do use them if you're interested in using the APIs programmatically, because it will give you easy access to them, and you understand it quicker, and you will be able to do integrate them into your programs. I want to mention Sebastian's poster, binary CIF. That's a compressed format for the main archival format, MMCIF. So please come and talk to him if you're interested in that. And that's about it. Just want to say thank you to, it's a huge team effort here. This is, we are at Rutgers, at UCSD and UCSF, and we are supported by NSF, NIH, and DOE. And I want just to leave this one slide in the screen. We have open positions, so so we have software developer positions and some postdoc position, and the director position is also available right now. So yeah, anyway, if you are interested in any of this, please come and see me. I'm also at poster D016 on Tuesday. Thank you.
So, yeah, thanks, Jose. So I have a quick question. So you talked about a structure summary. That's a new feature. Do you also have something similar that is grouped by UniProt ID?
Yeah. So this group summary, that I'm showing here, it's also available for, so groups are defined in different ways. One of them is sort of sequence clusters, but there's also a group that's a UniProt sharing the same UniProt accession.
So first of all, I'm going to be rude, the RCSB is a really tremendous resource, and I think everything we've seen depends on it. You've been traditionally conservative about models, and it's nice to see you've sort of accepted AlphaFold. How in future are you going to draw that line? So, for example, I don't think you've got the ESM 600 million structures in. How are you going to plan for that? Obviously, there are going to be lots more of these things, right?
Yeah, of course. And to be clear, we only have 1 million right now. We don't have even the whole UniProt collection, right?
Yeah.
We are planning to integrate more, and that's what we promised we would do, which is a very challenging thing because we are integrating the whole thing with everything that we have offered, like all the tools, comparisons and visualizations and search especially, which is very complicated. So we are planning to bring it all now, at least what's available right now. But yeah, the future is more complicated because things are going to happen. ISM Fold ISM Atlas or whatever they call it. That's one thing, but there's potentially billions and billions, and then if there are interactions, it's not really possible to do everything, of course. I think at the moment I can only tell you we are planning to do more, and so to continue on this path, but the future will tell us because whatever happens now, and there's AlphaFold 3 only like a few weeks ago. We don't know what's going to happen.
Okay.
Right, thanks, Jose.